Okay. Hello everyone. We are delighted to welcome Professor Lendert Gelens from KU Leuven, Belgium, as our IBS Biomed Colloquium speaker today. He received his degree from University of Bruxelles, where he studied laser dynamics and pattern formation with the research stay at in Spain and UC Berkeley in USA. And he then transitioned into biology as a postdoctoral researcher with the professor James Farrell at the
네,
Stanford University. Since then, since 2015, now he is leading the dynamics in biological system lab at the Department of Cellular and Molecular Medicine at KU Leuven. Okay, so please join us in giving warm welcome to Professor Gelens.
Thank you so much, Jay. Really appreciate the opportunity to talk here. And it's a pleasure. Thanks to you personally and to all the organizers.
Mhm.
Hi, everyone. Today, I would like to talk about biological oscillators, so systems in biology that cycle regularly, and how we can use experimental data and mathematical tools to figure out the rules that govern them. And the challenge is that biology is quite messy, so we can measure things, but writing down equations that describe what we see is not obvious. So the
Mhm.
question driving our work is, can we let the data itself tell us what the equations should be? And to make it more concrete, I want to focus on a particular oscillator that we work on in our group. And our favorite oscillator is the cell cycle. So life starts with a seemingly simple but really important step, one cell becoming two. And the cell first copies its entire DNA, and then those copies, here in red, get pulled apart by molecular fibers, microtubules, that are shown in blue, each half going to one side. And finally, then the cell splits in two. And that process repeats itself again and again with remarkable precision. And it has to be very precise because for most animals, around 30 rounds of cell division, they separate a single fertilized egg from billions of cells that make up a whole adult body. And so in our group, this is the system we focus on and understanding how it keeps time and what happens when that timekeeping goes wrong is important for development, growth, and disease. And to see the cell cycle in action across development, we need an animal where we can actually watch it happen from the very start. So
Mhm.
humans and mice that we typically use in biology as model organisms, they develop hidden away in the body. But fish, frogs, and salamanders, they lay their eggs externally. And those eggs are very large, so they're very visible, and sometimes they're also transparent in the case of many fish. So they're perfect for watching. And here I'm just going to play the video here of the salamander embryo. And one cell becomes two, then four, eight. And they divide very rapidly and very rhythmically. So it's very periodic, just like a clock that is ticking. And the embryo is not really trying to grow yet. It's just making copies and cleaving all of its cells, going from one cell to thousands of cells. And then something changes. The pace of the cell cycle slows down. The cells start doing something a bit more complex, moving around, they're organizing themselves, they're specializing, they're differentiating. And that ball of cells folds in on itself. And then after a while, a body plan emerges. You eventually get a head, a tail, organs, and so on. So the cell cycle changes throughout development, and it's that really early phase, those fast clock-like cleavages, those clock-like divisions in the embryo that we focus on in our group. And we do that because that's where the oscillator is simplest and it's most amenable to the kind of mathematical and data-driven analysis I want to talk a bit about today. And so this is Xenopus levis. It's the African clawed frog, and that is the model organism we work with most of all. And like the salamander that I just showed you, it lays its eggs externally. And what you're seeing here when I play the movie is one of those frog eggs going through its first divisions. And it's very similar to the salamander embryo. And I just want you to appreciate again how remarkable that is, because the egg is enormously big. It's a millimeter across. So compared to our cells, it's very big, roughly 50 to 100 times bigger in diameter than a typical human somatic cell. And yet it divides completely these big cells, and it does so very quickly, every 30 minutes. So that raises a physical question, because how do all the signals that control cell division coordinate across such a large cell? And also, how do they do it so quickly and so reliably? So the core molecular machinery that controls cell division is, I mean, the core machinery is also very conserved across evolution. So what we learn in these systems here in frogs and fish and so on can also translate to human cells. And I think this is where it gets really interesting with the Xenopus system, and perhaps also a bit crazy, because one of the powerful things that you can do with Xenopus eggs is that you can take their eggs
Mhm.
and you can put them in a ultracentrifuge, you can spin them around, and so you can pack the cells, you can crush them, and you can spin out the content in ultracentrifuge to take out the cytoplasmic fraction. That's what you can see here. So in the end, after several steps in the ultracentrifuge, you get this cytoplasmic fraction that contains essentially all of the molecular machinery of the cell cycle, but without the cells themselves. And so as we're interested in sort of figuring out the mathematical rules that underlie this oscillator, this is very useful to have, because we can now measure protein concentrations over time. We can add or remove components. We can perturb the system with a level of control that you don't have in a living embryo or any living cell. And it's a bit like taking the clock apart, the cell cycle oscillator apart, and putting all of the parts on the table, or in this case here in the tube, and ask ourselves, does it still keep time? And the impressive thing is that it does. So we can take that extract and we can encapsulate it into tiny droplets that are surrounded by oil, and we can watch what happens. So you can add fluorescent reporters to these droplets with extract, and you
Wow.
can look under the fluorescence microscope. And what you see is that every 30 minutes or so, all of
Mhm.
these droplets, they flicker, they pulse, and that's the cell cycle oscillator. It's the proteins that are rising and falling in concentration or activity, and it's the same rhythm that you normally see in the dividing embryo, but now in a tiny droplet of cytoplasm of frog extract. So there are no cells, there's no nucleus here. There's just a core biochemical oscillator, and that's really stripped down to its essentials. So what drives those flickering droplets? And it's a key protein called cyclin-dependent kinase 1, CDK1. That's the master regulator of cell division. And so when CDK1 activity is high, the cell divides. And when it drops, the cell resets and in interphase, and it starts rising again. And that repeats over and over again. And so what you're seeing here is exactly that. It's the activity of CDK1 measured over time in one of these droplets that I've shown you. And you can see that it's a nice periodic signal, and that's really the heartbeat of the cell cycle. And nicely is, or reassuringly, we can reproduce that. We have mathematical models for the cell cycle, and it's a system of differential equations. There are various versions of it, and they describe key biochemical interactions that drive CDK1 up and down, and it is able to capture not just the period of the oscillator, but also the shape. And so a quick detour, because when I said the heartbeat of the cell cycle, I actually did so on purpose, because this type of self-sustaining biochemical oscillator is something that we've known for a long time, and it's been described for a long time. Back in 1962, you had Dennis Noble, and he wrote down the first mathematical model for cardiac cells, showing how ionic currents could generate a spontaneous rhythm without any external trigger. So, and then he used those equations to fit also these experimental recordings. So far, we've been thinking about, or I've been describing a cell cycle as a purely temporal oscillator, something that ticks in time regularly. But what happens now if we take that same extract and we spread it out in a long thin tube of about 10 millimeters wide, which is what I'm showing you here. And the signal that I will show you is again the sensor for CDK1 activity.
Mhm.
So the CDK1 oscillator, if I play it, it doesn't just fire and pulse uniformly everywhere at once. Instead, it nucleates at certain points and it spreads out. You've got a wave of activity that travels through the cytoplasm and it sweeps along the tube. And this is the same biochemical oscillator we saw in the droplets, but now it just has a lot of space to move through. And at the moment, or in the moment that you add that space, and if you have a larger system, you get these traveling waves with a defined speed and a well-defined amplitude. And they emerge spontaneously from the coupling between the local oscillator and diffusion. And so for a one-dimensional system, as I'm showing here, I think it's really beautiful to watch. I can keep watching these type of things, but you can also look at it in two spatial dimensions, for instance, and that's what you can see here. And the dynamics get richer. In larger extract droplets, the same oscillator now generates these target patterns. So you've got concentric rings of CDK1 activity that expand outwards from a central pacemaker, a bit like ripples on a pond. But sometimes these wavefronts, they can also break up and you can get these spiral waves. So rotating, self-sustaining patterns that keep spiraling indefinitely and they're around a central core. And those spiral waves, they're a fundamental feature of excitable and oscillatory media, and they show up across physics, chemistry, and biology whenever you can have a local oscillator coupled to diffusion in space. And this is also why I made a detour to the heart and the cardiac system before, because these same spiral wave patterns, they also spontaneously appear and override the heart's natural pacemaker. And instead of a clean signal triggering a coordinated contraction, you get a spiral that can lead to disorganized contractions, inefficient pumping, and you get dangerous arrhythmia. But what I find beautiful to see in all of these systems, the extracts, the droplets, the Teflon tubes, the heart system, is that the same mathematical structures, they keep appearing. You get oscillators, traveling waves, target patterns rather, and spiral patterns. And that universality is really sort of nice to see and motivates a mathematical approach and also tells us that maybe we can use tools for discovering equations from data beyond the cell cycle as well and in different systems. So let me turn to that then. So in the end, after this sort of long introduction about the cell cycle, what we get from this by measuring the system is time series data. So we get measurements of one or more variables evolving over time. And what we want to do is get a model that can do some of the following things. First, forecasting, for instance. Can the model predict what happens next beyond the data that we measure? Second, we can look at interaction networks. Can one infer which variables talk to which other ones and how, which one activates which one and which one inhibits the other?
Mhm.
And third, can we learn something about dynamic solutions? Can we understand the full structure of the system in phase space, for instance? What are steady states? What does the limit cycle look like? How does the system behave under perturbations? And a good model, or ideally, it does all three of these things. And for me, an ideal model is a set of differential equations that describes how the system evolves, because that immediately gives all of these things. And so over the next slides, I want to show you some of these methods and also share one method that we developed ourselves. So before looking at the data-driven methods, I think it's worth appreciating how models have been traditionally built. So the classical approach is to combine prior biological knowledge and experimental measurements of individual interactions, and also combine that with scientific intuition to write down equations by hand. For instance, you know protein A activates protein B, B inhibits C, and so on. So you encode those interactions as mass action terms in a differential equation, and you estimate then the rate constants, for instance, from dedicated experiments or biochemical assays. And so this is also what Novak and Tyson did back in 1993 for the cell cycle. And the result is a system of many coupled equations. And I'm just showing 6 here. The system is much bigger than that. And each term is carefully motivated rather by known biochemistry. And that's really a nice piece of work. And models like this have taught us an enormous amount about how the cell cycle works. But you can notice what it requires. It requires a deep prior knowledge of the interaction networks. You need individual measurements of all of the rates, and you can need years of careful experimental and theoretical work to put that all together. And so here's a more realistic picture of the cell cycle oscillator, which is still much simplified. So, but still, you can already see that it's a web of proteins that are activating and inhibiting one another. And they're all coupled together in this very nonlinear network. And even this is a simplification, as I said. Many of these proteins, they also exist in multiple phosphorylation states, for instance, as I indicated here for a protein called WEE1. And each of these states can have different activities. They can have different activation interaction partners. So if you want to track all of that explicitly, the number of variables that will explode very quickly. And there's also another problem. Many of the interactions actually haven't been characterized yet. So we know many of the players and we know roughly which ones are interacting with which ones, but the precise rates, the thresholds, the nonlinearities, a lot of it is still unknown. So a fully mechanistic model would really require hundreds of variables and hundreds of
Mhm.
parameters,
Mhm.
most of which we simply cannot measure, I think.
Mhm.
So fitting such a model to data becomes very difficult. We've got so many unknowns and not enough constraints. So one idea is to not model every molecular detail, but instead measure effective input-output relationships of different modules in that interaction network. And so call that functional response curves. And extracts are really nice to do that, these frog extracts, because you can take the extract and you can add different inhibitors or different chemicals, and you can, for instance, stop cyclin translation entirely, and then add back precise and controlled amounts of non-degradable cyclin. And this lets us clamp the inputs of a module like this that I'm showing here. In this case, cyclin levels are the inputs. We can clamp them at a defined level, and we can measure the output of CDK1 activity in isolation from the rest of the network. And so was done in 2003, and I'm showing here the experimental results. And you can see that there's a difference in when you increase cyclin levels in blue and when you decrease cyclin levels in red. So there's hysteresis, and that points to the presence of a bistable switch in this system. And having such a switch makes the transition from interphase to M phase irreversible. And so what we did was to make or try to make a very simple or the simplest possible model for the cell cycle, keeping still as much realism as possible or experimental realism as possible. And so we kept these three players, cyclin B coupled to CDK1 and APC. So what happens is that cyclin levels rise. You've got mRNA of cyclin B in the frog egg. It's getting translated, and you're making cyclin protein. That protein binds to CDK1. You get this protein complex, and when it's in its active state, it will activate the anaphase promoting complex APC, which leads to the degradation of cyclin B via the proteasome. So that's a very basic core negative feedback loop. But then, so that's at the heart of the cell cycle oscillator in all systems. And then we use the fact that this cyclin CDK1 response curve is bistable, which was measured experimentally. And something that we noticed several years back is that APC in function of CDK1 is in fact also bistable. So this is also something that we can take into account. And we do so by capturing this essential nonlinearity of both of these switches with a simple cubic function,
Mhm.
which has the same mathematical shape,
Mhm.
or the same shape of the response curve, but far fewer parameters. And the
Oh.
result is a quite minimal
Mhm.
three-component model, three
Mhm.
coupled ordinary differential equations that is built directly from experimentally measured response curves that
Mhm.
reproduce the oscillations.
Mhm.
It tells you not just that the system oscillates, but also how these underlying switches can make the oscillations more robust. And so here's an animation of how that works with a single switch. And so you create you're going around this bistable switch, and you're producing these relaxation-like oscillations in the activity of CDK1 that drive the cell division in the embryo. And the way it works is you start making or synthesizing cyclin protein. So you move to the right, then you jump up in the bistable switch, then APC gets activated, cyclins are degraded, you move to the left and you drop back down and you turn around it. And it's a bit more complicated than that if you have both switches, of course. Okay, so I think that's a nice approach, and it still requires, but it still requires targeted experiments because you need to measure all of these functional responses,
Mm-hmm.
and you still need prior knowledge to decide which modules matter. So the question becomes, can we go further than that? Can we discover both structure and the parameters of a model directly from time series data without needing to dissect the network piece by piece? And so that's what brings me to these data-driven methods. And the idea is simple. You give the method a time series, so measurements of one or more variables evolving over time, and it gives you back a dynamical system, a set of differential equations that describes how the system evolves.
Mm.
There's no need to know the network in advance. You don't need dedicated experiments for each interaction.
Mm-hmm.
You just put data in, you get equations out, at least ideally, right? So I want to discuss a few of these methods. I will discuss shortly SINDI, which is sparse identification of nonlinear dynamics. It's a method that tries to find the simplest possible set of terms from a predefined library,
Mm-hmm.
and that explains the data. I'll also talk about symbolic regression shortly, takes a more exploratory approach. You search over um, all of the possible equation structures without a predefined library. And then there are also a lot of hybrid methods that combine these different ideas with neural
Mm.
networks, for instance, using the flexibility and the computational power of deep learning while still aiming for interpretable
Mm.
equations.
Mm-hmm.
And all of these methods share the same goal, to recover the function f here of the dynamical system,
Yes.
and ideally in a form that is both accurate and, of course, as it's a dynamical system, equation, just a functional differential equation, it's also interpretable. So you can analyze it also afterwards and learn from it. Okay, so let me start with SINDI.
Mm.
And that I think is probably the most, or perhaps the most widely used data-driven method for discovering differential equations from data.
Mm-hmm.
And I think the idea is really elegant. You start from your measured time series and you compute the time derivatives numerically. Then you build a library of candidate functions, for instance, polynomials, with cross terms, higher order combinations, basically all the terms that you think might appear in the equations. And then you ask which small subset of these terms, linearly combined,
Mm-hmm.
best explains the derivatives that you measured.
Mm-hmm.
And that last step is a sparse regression problem. So
Mm-hmm.
you're looking for a solution that uses as few terms as possible. And the idea is that nature tends to be parsimonious. So real equations don't usually have hundreds of terms, at least in physics. They have a handful of equations and terms. And the result is then a set of differential equations written in an interpretable symbolic form. And that really works very well on clean synthetic data. But what are some challenges? Well, there are three that I want to touch on, and they're important for biological data. First, there's noise. For instance, SINDI works by fitting derivatives computed numerically from the data, and numerical differentiation amplifies that noise. So small fluctuations in the measurements become larger errors in the derivatives. And as biological data is rarely very clean, this can be a serious problem in practice. The second one is sampling. So if you want to estimate these derivatives reliably, you need very dense and evenly sampled data. But biological oscillators often have also strong timescale separation, long slow phases and very fast short phases. And if you do equidistant sampling, you either undersample the fast dynamics or you end up with impractically large data sets. And in many biological experiments, having high temporal resolution is also very difficult. And third is then, whoops.
Mm-hmm.
And
Go.
third is model structure.
Uh-huh.
So Cindy needs you to define a library, and
Mm-hmm.
the right terms have to be in that library. For
Mm-hmm.
example, for polynomial systems, that's fine and it's easier. But biological interactions are also often nonlinear in waves
Mm-hmm.
that are hard to guess. You can have Hill functions, rational terms,
Mm-hmm.
ultrasensitive responses. And if your library doesn't contain the right functional forms, Cindy will either fail or give you uninterpretable, very high order approximations. So in conclusion, I think Cindy works beautiful in ideal conditions, but real biological data is noisy, sparsely sampled, and often governed by nonlinearities, which we don't always know in advance. And so that's a challenge, and there have been many extensions to Cindy which have solved various of these problems to some extent, but I think it still remains a challenge. So one way to get around that library problem in Cindy is to not define a specific library at all. So for
Mm-hmm.
instance, instead search over possible equation structures directly. And that's the idea behind this symbolic regression, where you've got candidate equations that are represented by these expression trees, where you've got mathematical operations like the addition, multiplication, the sign, sitting at these internal nodes. And you've got the variables or constants are the leaves in the tree. And then the tree encodes the structure of the equation. And you can manipulate it just like a living thing. And we can do two things. Typically, a crossover, for instance, which is what I'm showing here, it takes the two parent trees and it swaps subtrees between them. So you mix structural motifs from both parents and you produce something new. And you can have mutation,
Mm-hmm.
which replaces a subtree with a randomly generated new one. And you introduce some novelty in that way.
Mm-hmm.
And you can run that over many generations, and
Mm-hmm.
you keep the candidates that best fit the data, and gradually the population evolves
Mm-hmm.
towards better and better
Mm-hmm.
equations. So the advantage of over Cindy is that you've got freedom here. You're not limited to a predefined set of terms,
Mm-hmm.
and the method can, in principle, discover any functional form, including Hill functions and so on, and rational expressions. But it's computational costly, so it searches over all possible tree
Yeah.
structures, and it can also be sensitive to noise. So these two methods have complementary strengths and weaknesses. Cindy is interpretable, but it needs the right library, and symbolic
Mm-hmm.
regression is flexible, but it's computationally expensive.
Mm-hmm.
So we can turn to hybrid methods because, for instance, neural networks, they're very powerful, but they're not so interpretable. So you can try to combine them. And here's some three hybrid methods I want to touch on that I think are worth knowing about. So you can have, for instance, universal differential equations. They embed neural networks directly into a mechanistic
Mm-hmm.
model. You've got known parts of the system are described by explicit equations, and the
Mm-hmm.
unknown or poorly characterized parts are handled by the neural network. So you get the interpretability of the equations when you have prior knowledge, but the flexibility of deep learning where you don't. You can also do symbolic deep learning, where a graph neural network is used to learn interaction structure from data, and then use symbolic regression to translate that learned structure into explicit equations. And there's also Cindy with autoencoders, and that addresses an important limitation of Cindy, and that's the fact that it struggles in very high dimensions. So if you use an autoencoder that first compresses the data into a low dimensional latent space, and then you use Cindy to identify interpretable equations in that reduced space, you get a bit best of both worlds. So dimensionality reduction and
Mm-hmm.
symbolic discovery. And so all three approaches here, they share the same ambition, interpretable equations from data
Yes.
without sacrificing flexibility.
Mm-hmm.
And so we also try to develop a method like that, and it takes a different approach. So our approach starts from a different question,
Yes.
which is instead of asking what equations reproduce the time series, what
Mm-hmm.
we ask is what are the geometric structures in phase space that are consistent with this data?
Mm-hmm.
So here's a two variable oscillator, and you've got key geometric objects are these bulk lines here in green and orange. And they're curves in phase space where each variable's rate is of the rate of change is exactly zero. And where these null lines cross, you've got a fixed point. And so from time series alone, only the limit cycle here in white is visible, but the null lines are hidden. But they contain valuable additional information about the system. And that's what we try to learn using our method, which is called Klein, computational learning of null Klein equations.
Oh.
So how does it work? Here's the key trick. So instead of fitting derivatives directly, we train a neural network to learn the inverse relationship between variables. So you give v and a derivative of u as inputs, and it's meant to, and you train it on u. And so the network learns the full input-output mapping from the data, and that's the first step, to learn the relationship between the variables and their rates of change across the phase space visited by the trajectory of the limit cycle.
Oh, so Kellen, I'm sorry. So
Yeah, sure.
here, anyway, V and U, derivative U is input and then output of
Yeah,
this will be
yeah.
U, right?
Exactly.
Okay,
So
okay,
it's
gotcha,
the sort
gotcha.
of opposite of what you typically do. And so I think it
Okay,
will become clear in the next step why we
okay.
do this. Because in the next step is once the network is trained, we simply set the derivative input to zero, u
Mm-hmm.
to equal the derivative of towards time is zero, and ask for each value of v, what is the value of u that satisfies that condition? And so
Oh.
the answer is then the null line.
Mm,
So we go from time series to a geometric curve in phase space, and
mm,
not by regressing on the derivatives
mm.
directly, but using them as inputs to learn a smooth mapping. And that makes the method more robust to noise than Cindy, for instance.
Oh, wow.
So we demonstrate this here on the Fitz-Nagomo equation. So it's a prototypical relaxation oscillator, and it's built on a bistable switch as well. So it's very similar to the cell cycle oscillator we started with. And here I'm just showing an animation which shows the training loss. So as we train, the network gradually separates the limit cycle from the null lines and it converges on the correct geometry. So here's how it learns both the limit cycle and the null lines.
Wow. Oh, okay.
And so I think that's quite useful because you now get a clean curve that
Mm,
describes the different null lines and they're sampled densely across the phase space.
mm,
And you can now fit an equation, and fitting an equation rather to this type of curve is a much simpler problem than fitting equations to noisy time series.
mm,
So this is where we then connect back to Cindy and symbolic regression because we feed the learned null climb points into Cindy or symbolic regression or Pizer, for instance, and ask them to find the simplest equation that fits this curve, including strongly nonlinear terms like Hill functions and rational expressions. And the methods like Cindy and symbolic regression would have difficulties doing that from the time series alone, but doing it for the null clients is much easier. So the optimization problem is now more geometrical rather than dynamical. And there are no timescale separation issues. There's no
mm.
amplified noise from sparse regression on derivatives. And then afterwards, once we have the null clients, it's a much easier problem to get the final equations out. And so to test Klein on real experimental data, we need a system where we can measure oscillations cleanly and at a high temporal resolution and with low noise. And biological data from the cell cycle is still is more difficult, so we're working on that. But so what we do is we turn as a proof of principle to the chemical oscillator, the Blusov-Zhabotinsky reaction. It's a chemical solution that spontaneously oscillates and it changes color periodically as the concentrations of key species rise and fall. So there are no cells nor biology, it's just no biology, it's just chemistry. And we can measure that with high precision over many periods. And I think it gets more interesting because if you spread the BZ reaction out in a dish, you get the same spatial patterns we saw in the cell cycle extract, target patterns that expand radially outwards and spiral waves that rotate And that's very similar to what we see in the frog egg extract and in the cardiac tissue. So again, it's an example that you have the same math, but different chemistry. And that universality is also why the BZ reaction is, I think, a powerful test bed, chemical test bed for biological systems. So here's the pipeline applied to the real experimental data from the BZ reaction. So in the first step, we've got the raw measurements, which look very clean. So we've got time series of two chemical species, a cerium ratio and a bromide ion concentration, and they oscillate with characteristic relaxation-like way. And then in the second step, we clean and pre-process it, although the signal is already very clean to start with. But we normalize the signals, we align them, and we prepare them for input to Klein. And then we apply Klein, and we outcome these null Kleins in orange and green. So you've got the two curves in phase space that encode the geometry of the system. And it works quite well. So Klein recovered this hidden geometric structure of the oscillator directly from the experimental time series data. And it handled strong timescale separation and nonlinearities that would cause, for instance, Cindy or other methods to fail.
Yes.
But the question is still, how do we know whether these null Kleins are actually correct? So we need something to compare it to, because if we just have the time series, we have no idea whether it's correct or not. But the nice thing is for the BZ reaction is that it's one of the best studied chemical oscillators in history. So back in 1972, you had Field, Karush, and Neuss who worked out the full reaction mechanism, 21 interacting species. And in 1980, or in the 1980s, John Tyson reduced that big system down to just two interacting components, which is called the oreganator, and it's a landmark model in nonlinear dynamics. So the top right shows the phase space and the null Kleins from Tyson's reduced oreganator model. And the bottom right shows what Klein produces from these raw experimental data. Without prior knowledge and no reduction, just time series in, you get the null Kleins out. And I think the agreement is quite nice. So Klein has rediscovered from data this similar geometric structure that took a very long time of chemical analysis and theory to work out. So the BZ reaction, it gives a proof of concept that Klein can recover null Kleins from actual experimental data and match it to a sort of ground truth model derived from decades of careful analysis. And I think the question now is whether we can do the same thing for the cell cycle, but it's harder, of course, because the data is more noisy, fewer time points, and more unknowns. But I think the extract system that we have is giving us a nice system to pursue that question because we can measure many oscillations over many different conditions and we can perturb the system in controlled ways and watch how the oscillations change. And so I want to show you some of these things before I wrap up. So it turns out that the extract doesn't just oscillate biochemically, but it really reconstitutes the full physical machinery of the cell division. So in these droplets that I'm going to show you here, you can watch here in actin networks that are assembling and disassembling. So you get like spontaneously driven by the cell cycle oscillator, you get an actomyosin ring that is oscillating and pinching off. So if you actually had a cell membrane, you could think that maybe this is going to divide the cell in two. If you look at a different signal for microtubules, what you can see is that spontaneously, periodically, you get these microtubule asters that are formed and polymerize and depolymerize. And if you add sperm chromatin to the system, what you can see is that actual nuclei emerge and form, and you have got nuclear embryo breakdown as well. And that also is driven periodically by the CDK1 oscillator. I think that's quite remarkable because you've got this droplet of cytoplasm and it really reproduces the rhythmic physical organization that drives a cell through division. And so we can start measuring how CDK1 changes over time under many different conditions. So what happens if you have microtubules and you inhibit actin, or vice versa, you have actin, but you inhibit microtubules, you keep both. So you can start playing with the mechanics and the spatial structures in these artificial cells, in these extracts, and see how does the shape of the oscillation change. So you've got a CDK1 oscillator, the basic oscillator, and see how do the oscillations change if you change the different conditions of interactions with spatial structures, for instance. And apart from only measuring CDK1, we can also measure other things. We can measure, for instance, on top the actin signal. So we can see how actin behaves over time, or we can see how microtubules change over time, or so how these signals change over time. So what that gives us is that we have a time series of different time series of a single experiment, both microtubule signals, actin signals, CDK1 signals, but you can think of other things,
Mm,
anything you have a fluorescence reporter for, and they're oscillating together under multiple conditions. And so we can feed that into data-driven methods.
mhm,
And the goal would be to get a minimal dynamical model out. So a small system of differential equations that describes how CDK1 and microtubules actin interact with each other and drive each other.
wow.
So not a model that we wrote down by hand,
Mhm,
because we don't have prior knowledge or not that much prior knowledge about how
mhm,
all of this works, but just let the data itself lead us.
mhm,
And then we can analyze different mechanisms. So that's work in progress. So we've got a lot of these data, and we've got a method, for instance, validated on the BZ reaction, and now we're trying to go further with that. And I think that leads me to my conclusions, where I would say that cell cycle is a beautiful oscillator,
mhm,
and cell-free extract give us a really clean experimental window into its dynamics across conditions and at multiple levels. You've got classical data-driven methods like Cindy, for instance, that work really well under ideal conditions.
mhm,
They still struggle with noise, time scale separation, and unknown nonlinearities that are typical for biological data. And I introduced Klein, which offers a new approach targeting the geometry of the phase space rather than fitting time series directly.
mhm,
And that allows us to recover interpretable null-Klein structures from experimental data and combine it with symbolic methods to extract equations.
mhm.
And so we validated that on the BZ reaction, and we were hoping to get something new out for the cell cycle as well. And there are a lot of interesting things that lie ahead.
Mm,
Um, for instance, well, exploring many other methods, but you could also think about going to spatial systems, so moving from ODEs to PDEs. So I've showed you a lot of traveling waves and spiral patterns in frog extract and the BZ reaction. So we can apply methods that recover these equations from imaging data. I think that would be really nice. And there are methods out there, of course. Second challenge is missing variables. How do you deal with that? Because in most biological experiments, we can't measure everything. So we're also looking into, for instance, delay embedding techniques that offer a way to reconstruct hidden dimensions from
mm,
partial observations. And well, I think also the landscape of all of these hybrid methods is changing fast. You can combine a lot of interesting methods with one another to improve the different methods. All of them bring something else, and I think understanding which tool to use and combine them is still has still a lot of options. And so I want then to thank everybody in the group. I think a lot of the work that I presented on data-driven methods was done by Bartosz Prokop, who was a graduate student in the lab who finished his PhD
mhm,
about half a year ago. Experiments with extracts have mainly been done by Martina and
mhm.
Lili Piñeros, and data analysis on experiments mostly by Nikita. And so here are the current lab members on top. And so Lily and Bartosz, Lily is currently in UC Berkeley with Rebecca Hield, and Bartosz is currently looking for a job in industry. And so I want to thank all of you. And if you're interested in knowing more about this, you can visit our website here on the bottom left
Mhm.
and find some more information there.
Great.
So thanks so much.
Thank you. Wow.